So thanks so much for having me there. Before I start, I should also say I like having discussions. And at some level, I'm trying to push you a little bit with my talk. And I want you to push back. Like, let's have a real discussion about those issues. And this talk is at some level an extension of the microprocessor talk, the microprocessor paper. Microprocessor paper is almost a little cliche by now. So what I'm trying to do this time is take pieces of that and now relate it more to biology than what we did there. And much of the talk will be a rant against the way how neuroscience is trying to use tuning curves to understand how the brain works. But we'll get there. So let me start with, when I started as a PhD student, I somehow found neuroscience incredibly exciting, but I wasn't quite good at saying what I meant with that. And it took me a long time to understand what those questions were that were really driving me. And there's many questions, and there's many good questions in that area. Do I have a laser pointer or something? I'm sorry. What it means to understand very strongly depends on which hat you're wearing. If you're in the mindset of a medical doctor, it might be that understand means that you can fix it if it's wrong. At some level, you understood it. No, if you can fix it, the problem's solved. If you understand it deeper than that, if there's this disease, I need to give that drug. That's kind of an unnecessary luxury. There's also another view where you can say to understand means that you can simulate it. If I can take the neurons in Konrad's brain and have a function that describes all of them, and I simulate it, and it talks like Konrad and insists on the same weird ideas as Konrad, at some level, it feels like some kind of an understanding. We can build it. And then there's, of course, the classical 3 levels of MA. There's the computational level, where you can say, the question that I might have is, can I find what that problem is that the brain is solving and understand behavior as optimally solving that problem? And so it's basically the question, what is that the brain is trying to optimize? There's the algorithmic level where the focus is more on what are the variables and what are the transfers between them. And then there's the mechanistic level. Now, what all these share here, and at some level this one as well, is one measure of success is that we can describe a lot of data. But each of them have something extra on top of that. The computational level says, on top of describing data, I want it to be in terms of what it is, what the objective is. And at the algorithmic level, on top of describing data, I want to know about, I want to have a description of what the kind of data and transformations are. And at the mechanistic level, I want to describe how the microscopic things in the brain make these things happen. There's a 4th class. I'd call them nihilists. Ma never talked about them, which is you could just say, forget about that extra something and just say understanding is you can fit a machine learning system to it that describes the spikes and is really good at making predictions of spikes that you have otherwise. So there's, in that like deeper understanding domain, there's at least 3 of the 3 traditional Ma types of questions, and then there's the nihilist one. And at some level, the bulk of good modeling work subscribes to one of these. And a lot of the wrong disagreement between neuroscientists is that people who have different drivers for themselves disagree with one another. And if I'd get a drink, a free drink for every time someone mechanistically attacks a Bayesian model, I'd be drunk for the rest of my life. No, it's so often I hear that, but how could the brain be Bayesian? Couldn't it be dopamine or something like that? No, that's not even a meaningful question. So there's different kinds of understanding. And at some level, on different walk of mine, I can wear each of these hats. But at some level, when I started neuroscience, I wasn't clear which of these hats I was wearing, and in fact, I was constantly mixing those hats up, and I was confusing myself about it. But there was like that dream that I could, at some level, understand the whole brain in a way that would make sense for me. And there's multiple ways how that dream works. And I want to talk with you about that dream. And then I want to make that dream a little worse. So one idea is that successive tuning curve mapping could help us and could allow us to understand the brain. And if we read Hubel and Oisel, this is where it starts. You can say the retina has receptive fields that tell us if a point is bright relative to the vicinity, and primary visual cortex tells us if there's little bars relative to the vicinity, and then V4 might deal with little curves and colors, swirls, and somehow then once we go further up, we will have objects, and once we go further up, we will have decisions based on those objects, and this is sort of this is what today I will mostly try and poke holes into that. But that is certainly for me when I started it. That felt very real, that framework. And we'll see how real it is at its promise. Now, a lot of people realize that that won't quite be enough. And so what are the backups then that I think a lot of people start thinking about the brain that this is what leads to understanding how it works. Then a backup, if people say, well, that understand the brain based on tuning curves won't quite work, is that they say local reverse engineering. What we could do, we could look at each neuron, we could measure its inputs or set its inputs, maybe do optogenetics on them, and do systems identification. It would tell us what the spikes are as a function of the input. And at some level that can lead, if the tuning curves won't lead to a causal understanding at some level, that should. And I should briefly mention this dynamics approaches. You can alternatively fit dynamics. And it might still be that this doesn't give us enough data. So a lot of people say, well, if neither of them by themselves can work, well, if we on top of that have anatomy, it can work. And then some people go a step further, say none of them, even in combination, can work at really satisfactorily answering our questions. We really need theories on top of that. So let's take the tuning curve approach. But what I want to use as a guiding idea for the rest of the talk is the microprocessor paper. And for those people who read it too often, I apologize. Here's my favorite nervous system to study. This is the MOS 6502. It's a classical microprocessor from the 70s. It has 3,000 transistors. You can, if you take a class in computer engineering, you can relatively easily understand it. In fact, if someone explained to you how it worked and you were given a few months, you could probably build one yourselves. And the cool thing about that is that we really know how it works. So we know about the pieces that we have on the chip. There's the memory interface that gets data from the main memory into the chip and back. We have an instruction fetch circuit that gets the correct next instruction. An instruction decoder that takes the instruction and converts it into a bunch of on versus off lines. And then we have registers, which is its internal memory, so that it has memory. And it has a math unit. And ultimately, the instruction decoder sets what the registers and the math unit do, and then it's being fed back onto the registers and can, through the memory interface, be put back onto the main memory. And the interesting thing is we understand this circuit at every possible level. So we can zoom into that, say zoom into the math unit. Here we have a 1-bit adder, has 3 inputs, the 2 bits and a carryover, and it gives us an output bit and also a carryover. And then we can zoom into that further, and we can say we have logic primitives, AND gates, XOR gates, OR gates. And ultimately, they consist of transistors, and the transistors have a hardware implementation there. So now you can say, well, why am I doing that? Why do we need to look at the microprocessor? We have brains. Can't we just study the brains? So here's the problem with brains. If I say something stupid about the brain, no one can call me really out on it. They can, maybe I disagree with some data, but that's the only way how people could disagree with me. The problem is that I may be very wrong, and still no one can call me out on that. And a little later we'll get how experiments make that very easy that happens. But the problem is that I can't know if I'm right or if I'm wrong. The great thing about the microprocessor is we can know that. And now, a microprocessor, let me briefly say something about behaviors. Just like with animals, we need to study behaviors. For that, we have 3 behaviors: Donkey Kong, Space Invaders, and Pitfall. 2 popular behaviors that are being used many times there. Now, how is the microprocessor like a brain? Well, the microprocessor consists of a lot of small elements, transistors, with exponential characteristics, which arguably neurons have as well, if you use Poisson models. It's recurrent. It's nonlinear. It reuses part of its circuit to solve many different problems. In those regards, it's similar. In lots of ways, it's dissimilar. No, neurons probably have real noise, and this doesn't have real noise. Neurons have thousands of inputs, and the typical transistor is a very small number of them. But none of these factors really should make us believe that it's easier to study the brain. It should be harder to study the brain. Now, what's cool here is we can now take that system that we know how it works, whenever we do a study, we use logical assumptions. We say, this is how we interpret the result of a given experiment. Now, what we can do here, which we can't do with the brain, is we can say, well, this is the experimental assumption, that's the data analysis assumption that we put in, is the result that we get correct. So let's take the first thing. The most obvious thing that people do with brains is they do lesion studies. So we have a little being, 3,000 transistors. It's less like a mouse, and maybe it's more like C. elegans, but that's a detail. And what we do is we go and we lesion transistors one by one. And the only thing that we test is, does it get through its boot sequence? And there's a whole bunch of transistors that are just lethal, lesions that are lethal. There's plenty of brain lesions that are lethal too. And then you find some transistors that only make one of the games not boot. So what would we conclude if we use the typical logic that neuroscientists use? We might say, well, look, here's kind of a Donkey Kong region. Look, this is kind of pretty pitfall heavy in that area. We might be inclined to draw such conclusions. Now, we know the microprocessor, and we know that the idea that this is a Donkey Kong region is entirely unjustified. And at the same time, this is something that we also know with lesion studies. At some level, all the papers that come out that do some kind of a lesion and optogenetic inactivation study, there's always somewhere a sentence that is a nod to, we can never be sure about what that means, but that's not how it works in our brain. If we inactivate a brain region, we have a loss of a function, we will very strongly be willing to assign causal meaning to that, where we say this brain region therefore solves that problem, that it can no longer solve once we lesioned it. And it just nicely shows what the problem is there. And there's a beautiful philosophy paper that says, are necessary and sufficient, necessary and sufficient. So the typical way how we ask questions in that domain is that we ask a necessity statement. We take out something, a cell or a subcircuit or a type of cell or a brain region, and we take it out. It's not there anymore. And then in some rare cases, we have a sufficiency statement. We take some piece of the brain, we inject current there, and we find that then maybe the animal moves their hand to a certain position. But both of them are very hard to interpret, because if it was a linear circuit and all that, then the interpretation would be much easier. But here we have a nonlinear recurrent circuit, and the effects can be arbitrarily irrelevant. Okay, so now you can say, well, we didn't use specific knowledge about the microprocessor. Now, the microprocessor, we know a lot about the microprocessor. We could have used it, we didn't use it. Could we have done better if we had used it? And yes, we could have at some level. If we built in that we know that there's a signal, that there's an instruction decoder, we could have done better. Now, as long as the system is nonlinear and recurrent, it's very hard to rescue it logically. And I should also say neuroscience doesn't. So it's not if we have these studies that they really combine a lot of data that is meaningfully orthogonal to really use prior knowledge from the brain to interpret the lesion data. It's very hard to have such data. And but now let me quickly get to the tuning curve. So we can also have spikes there. We recorded from, we have, of course, full recordings from all transistors. That's the nice thing. We have a great simulator for it. So any experiment you can possibly dream of, you can do perfectly on the microprocessor. And we just arbitrarily declare the off-to-on transition a spike. And here we have our spike trains. And if you go to a physiologist, I went to cosine and passed it along. Which brain region do you think we recorded from? A lot of people think that could be basal ganglia. I don't know why that is, but when people look at it. No, it looks quite a bit brain-like. It looks a little epileptiform if you zoom into that, where like certain things appear they are repetitive, but not so much. Tuning curves, okay? Now that we have that microprocessor, we can record from a couple of transistors. And we can see, are they tuned? In that case, we take the brightness of the last written pixel. That sounded like something that an animal that spits out rapid decisions about brightness. That could be a good tuning curve. And indeed, we find a bunch of transistors that very strongly have firing rate that depends on the luminance. Now, the way by default as neuroscientists we'd interpret that is that we'd say, oh, we have strong tuning to luminance, therefore we must have luminance encoding. The cool thing, however, here is that we know what the role of the transistor is. We know what the meaningful description is, what they do. And obviously they're not. Up there we have instruction decoding. Here we have a register. All of those transistors, we know what they're doing and why. And it just shows the caveat of just because you find a tuning doesn't mean that it's in any that the role is in any meaningful way characterized by that. And that's a little bit a problem if we want to build causal stories out of it. And at some level, what we mean when we say we understand the brain is that we can tell a causal story. That brain area encodes that information, and that next area does that with it, and so forth. That's the story that we like to tell. Okay, now let's look at what the assumptions are. So I'm going to spend a lot of time now thinking about tuning curves and what the assumptions are for us, because intuitively it makes a lot of sense. You know, that the idea that we have that hierarchy of processing, we have tuning curves, we can understand what the role of a neuron is by looking at the tuning curve. That seems very natural to us. So what are the implied assumptions? So the first one is that we can guess the dimensions. So a neuron is nonlinearly related to things, potentially nonlinearly related to things in the outside world. And if we are to have a meaningful tuning curve, for the whole concept of tuning curve to be meaningful, we must be able to figure out what the dimension are that the neuron cares. And that comes with an implied assumption that all other dimensions don't matter. That this is what the neuron cares about. There's some dimension and some high-dimensional space. All orthogonal directions are irrelevant for that. And the question is it realistic that humans could guess that dimension? And if they can't, the whole framework is problematic. Now, what we do is we put an electrode, and that's what I did as part of my PhD. I never published because I'm a horrible experimentalist. But I put electrodes into primary visual cortex and recorded from the neurons there. And I gave some stimulus. And underlying for everything is that we can guess what the right dimension are. There's also the implied assumption that all neurons will share that. Because what we do experimentally, we take a small low-dimensional stimulus set, typically. And there's exceptions, but we typically take a very low-dimensional space. Maybe it's color stimulus, where we just change what the color is. Or a brightness stimulus, where we just change what the color is. We need to be able to guess it, and it needs to be conserved across neurons. And then there's also the assumption that it must be almost linear. Even if it did care about that one dimension, and it was a horribly complex nonlinear function, the tuning curve that we can draw from it wouldn't meaningfully characterize what the neuron does. And there's also an implied assumption that each neuron, we know what the feature set of its input are, and then we look at what it does with those. And if that assumption was true, then everything would work just fine. No? If we can describe all of the retina as a couple of localized receptor field, and all of V1 as a couple of oriented edge detectors, in that case, that framework does work. We can reverse engineer one layer, and then we go to the next one, and it works like that. And that's at least how it feels like to me, how it felt like. Okay, so now let me first, can we actually guess the dimensions? I think we can't. It's hard to be sure, but here's just some examples. This is from my lab. Primary motor cortex, we all believe that it encodes velocity, x and y velocity. But it actually cares an awful lot about reward when we recently checked. There's older work in V1 where expectations matter, cognition. The cerebellum, the belief was for a long time that the cerebellum is really boringly just correcting movement errors, and it's involved all over the place for cognition. So when people look, they find that. And here's like a nice example. So this is joint work with Rob Turner, who recorded from basal ganglia. And it's an interesting, complex task. So the monkey always starts in the middle, and then the monkey moves into 4 different directions, and it's a sequence. So it's like go left, go upper left, go lower left, go to, and so forth. And the monkey does that many times. And there's different sequences that are cued in different ways. And the interesting thing is there's many things that in principle neurons could care about in the movement system. This is the part of the basal ganglia where we expect movement. Well, and we find all of them. And so on this axis, we have different regressors, different things that the neuron could care about. On this axis, we have how many of our 100-some neurons actually did care about that. And spatial cue. There's some neurons that are more active if we give a spatial cue. There's certain other neurons that are extra activated if there's a color cue. There's certain neurons that care if there's a center cue. There's some neurons that care about the first element of the sequence, second element of the sequence, third, fourth, the end of the sequence. Some neurons care about the efficiency of the movement. Some neurons care about the direction of the reaches. Some of them care about the kinematics. And a whole lot of them care about the reward. But the important thing is any meaningful regressor we tried here describes significant variance to a whole bunch of neurons. Now, that doesn't prove anything, what I'm showing you here. It's because it could be that there's just one dimension that depends on all 12 or something of them. And we just can't guess it. Once we would have guessed it, it only cares about that one. But that seems very unlikely. And in fact, if you look at different neurons, they weigh all of these in different ways. Some neurons don't care at all about a good number of them, and then very strongly about a few. Some neurons care about a whole bunch of them. So, and this matches it when I ask my physiologist friends, where I say, well, have you tried extra regressors that could be relevant, maybe attention? Will basal ganglia care about attention? Like, you bet. So I think anyhow, I would speculate that the bulk of halfway meaningful extra regressors significantly improve predictions. So if you take neurons somewhere, you brainstorm what they could care about, you will keep finding things that they care about. So what that means is that I believe that the evidence for that idea that it's a simple space and that we could realistically guess it and that it's shared across neurons, that's just not true. And that's the first argument. Let me make a slightly different argument with a different data set. So this is a collaboration with Matt Smith. We're currently writing that up. What we did is we had neurons recorded in area V4. It's an area that's known to relate to color. We don't know if it only cares. I mean, we know that it doesn't only care about color, but we don't know how broad, what all the things are that it cares. And then what we did is we stimulated it with different colors. So color is a circle. This is just the angle on the color circle. And here we have the activity. It's scaled so that we can see things better. And let's first look at the black curve. We show a whole screen color stimulus in that case. And we find that the neurons, these are just 2 example neurons. It's true generally, what I'll be showing you. We find very nice color tuning. So this neuron has very low activity if the stimulus is yellow. It has pretty high activity if it's like violet or green or bluish green. That other neuron here is very active if it's red and little active for other colors. So very strong color tuning, as we should expect here. But now if the concept of color tuning is correct, we should have a generalization, which means that if it likes red in artificial scenes and its coding is red, then it should just as well like red in natural scenes. So what we did is we know what the spatial receptive field of the neuron is, and we can extract the dominant color in that area. And then we can just as well plot the firing rate here as a function of that color. And I should say this is a model fit. It's somewhat smooth relative to what we have there. And in green, you see that response to that natural scene. Well, all of a sudden, that neuron actually here likes blue, and it's a little complex. It likes violet or something here. And the interesting thing is, across neurons, there's not even a correlation between the preferred color. So what that means is, again, it's our guess that color is the right space. If color was the right space, those 2 curves should look the same. But they don't. They look entirely different. So again, this is just the idea that there's a simple space that we can guess isn't supported by this. Okay, so that's the first part. So I'm saying we can't guess what the right space is. And on top of that, the neurons don't even lie in the same space. Now, the second one is to which level are they linear? Like, for the logic to make sense, we need to have that in the right feature set there, they should be linear. If they're not, then the whole analysis breaks down. So what we have here is we have a single cell. And what we did here is we did machine learning. And we did, there's a web page called Kaggle where people run machine learning competitions. And this was the maximally purposefully non-creative project. We used the perfectly standard Kaggle pipeline that people use for a lot of other things. How does that work? We take, so first GLM, this is what we typically do. I wrote a lot of papers about cleverly fitting GLMs to neural data. And pseudo R squared, what we have on this axis, is just a measure of how good we are at predicting spikes. And the GLM predicts some spikes. It's very highly significantly predicts spikes. It's a super significant tuning curve if you want. And then what we can do is we can take another model instead of the GLM. We can take a neural network. And it does better in that case. And we can use XGBoost, which is a very boring machine learning technique that probably wins most competitions of all of them, which does better than neural networks. And then we can use a so-called ensemble method that takes all these models here and combines them. And it does considerably better. Now, what do we see here for this one neuron? And it's anecdotal. On the next slide, I'm going to show you the data for all of them. So the GLM is effectively a linear technique. It prevents neurons from having negative spikes, but otherwise it's in first order it's a linear technique. And it describes some data, but it's dramatically worse at predicting spikes than modern machine learning. What that means is that a linear model, which is what the GLM does much worse than the nonlinear model. Again, pointing towards a violation of this idea that it's simple and linear. It's not linear. And you get a lot of mileage by doing nonlinear modeling. Now you can say, well, that's just one neuron. Who cares with one? And I should say all the code is on GitHub if anyone wants to play a similar game on your own data. Here we have the data for all neurons. It's a little crowded. On that axis, you have the GLM pseudo R squared. Again, pseudo R squared is just a measure of how good you're predicting spikes. That's the modern machine learning, pseudo R squared. This line would be where the two of them are identical. And you can say the pseudo R squared is double, maybe something like that. And so this is the mean plus minus the 95% confidence interval across neurons. So you're getting about twice the pseudo R squared by using those nonlinear techniques. So neurons are rather strongly nonlinear. Okay, cool. So now what I wanted to tell you about tuning curves so far is that the way we approach it, all of our underlying assumptions don't work out. They aren't linear. We can't guess those dimensions. And at some level, therefore, we shouldn't hope that we can understand how the system computes based on that set of approaches. I'm not saying that it's not interesting or important. Of course, we need to know which neurons in which brain areas, what roughly the things are that get them to fire, but it won't be enough to really understand how it computes them. Great, let's go to the next piece. LFPs and power laws. We have the microprocessor, we have those regions. We have all the transistors. And now you can say in neuroscience, what we often use is we use LFPs where we have a relatively low impedance electrode that's big that picks up the signals from many electrodes. And we can simulate the same thing on the microprocessor. So we have spatial averaging here from those 5 regions. And this is how they then look like. Now, the first thing is it does look a little epileptiform because you do see the clock signal in there. But it also is multi-scale. And in fact, in good approximation, the power spectrum is 1 over f. 1 over f power spectra, we know in neuroscience that it's a sign of self-organized criticality. Not sure how we should think about it here. And we can do Granger causality. We take those 5 signals, just the way you would if you recorded LFPs. And we can fit a Granger causality model through that. And so through that, we see that there is a subnetwork that is basically the resting state network that's always at some level involved here. And then there's a modulation that is task-specific that is on that. And as neuroscientists, that's how we would, by default, interpret it. We would say, well, there's this brain regions. They are all coupled. And at certain points of time for certain tasks, then that region starts talking with that other region. But we know that the anatomy here is entirely fixed. And we also know that the network, some of the connections are real. They're real on the chip. Some of them are not. And it's not really clearly better than chance. But it looks really good. And you could have, if this was the brain, you could have speculated a lot on what those results mean. And I would probably have, if I got like that same data here from a brain, I run those algorithms, I would certainly, and I get those results, I would probably be writing something about those Granger causality graphs. Very hard to interpret. Okay, what else do we have? The nice thing with microprocessors is that we can record from all neurons at the same time. Because there's just 3,000 of them, and it's a simulation. So we have all the data. So here we have recordings from all of them. Here we have time on that axis. We have the 3 different behaviors. And each line is the activity of a transistor over time. No, great data. Also, really noiseless data. There's no Johnston noise on our recording electrodes. It's by every definition, this is a perfect experiment. And now we can run non-negative matrix factorization, which people often do when they use such data sets. Now, hey, we are finding something really meaningful. We find two signals here, dimensions 1 and dimensions 4, that are related to the clock signal. And in fact, why two of them? It turns out that those old microprocessors have some activity that is time-locked to the clock transition to the upstate, and some other activity that's time-locked to the downstate. So it makes sense that we'd find two of them. We also find signals that are related to the read-write signal. So great. We do find something that is real. Now, is that what we mean when we say understand? No, it's like this is real data. This is a real part of it. But this isn't sufficient to allow me to claim any kind of understanding. And in the same way, we do find in neuroscience many times that we have data that is real, there's a real correlational structure between them. The question is, how much do we really learn about the nature of communication in the system from it? And now, let me go back to what we can do. Now, so far I've frustrated everyone. Everyone's looking really sad right now. Let's see what we can do. So the first thing is that we can do generalization studies. And I think that that's super important. It's very doable. So let's say you do an experiment. You measure a tuning curve. That is usually where your paper ends. You measure the tuning curve. You come up with like some funky theory. If it's a modern paper, you might even collaborate with a theoretician who builds you a theory for that. But that's where the paper ends. What we should be doing is run a generalization study. You measure color tuning curve. Well, let's just redo that in the context of natural scenes. Or let's redo the attention experiment with a slightly different set of stimuli. Because at some level, an experiment where you find something without a generalization study means nothing. It's a local observation. The generalization always comes in our mind when we read a paper that measures a tuning curve. And that can be a very clever tuning curve. It can be a tuning curve relating to the log probability of the decision being right or something like that. But even then, unless we show that it generalizes somewhere, it really means preciously little. Because if we want to make the point, this neuron encodes the probability that the signal will be to the right, well, then we'd want to know that it does that across a couple of different situations. It's very rare that people do generalization experiment. And from my own experience, I can at some level say why that is the case, because all my generalization studies fail, almost all of them, both in psychophysics and when we do neural data analysis. It's just you have the data, you have like beautiful model, makes sense, and then the generalization doesn't work. And it has often happened to me. I should briefly mention in Bayesian psychophysics, there is a trend where people start doing that. So McLelland has very much been pushing for doing generalization studies. And there is a movement in that direction. But it's something that would make science really would make neuroscience a lot more useful. Now, at some level, I poked some holes into the assumptions that are underlying this idea that if we have enough tuning curves, we can ultimately understand how the brain computes. Let me go one step further. So as I said, like the backup solution, if you then tell people tuning curves are programmatic, is that they say, well, okay, tuning curves are programmatic, but on top of tuning curves, we could measure how a neuron relates to its inputs and outputs. And so let's see if that is possible. So if we have tuning curves, we have a model that basically models the number of spikes that we have as some function with lots of parameters of the stimulus. And that function could be based on deep learning or whatever we put in as regressors here. That is the logic of that. And now you could say for that reverse engineering idea, you can say really the spike should be a function of maybe the stimulus is still necessary, but the past spikes of other neurons. No? Like as soon as I'm not just a sensory neuron, my activity should only depend on the past activity of other neurons and myself. And you could say, well, may that be possible? Well, here's the problem. The number of spikes that we get in one experiment almost never exceeds 100,000 spikes. Anyone in this room ever recorded more than 100,000 spikes? Yes. Okay, but it's very rare that someone gets 100,000 spikes. Now, if we want to identify a neuron based on its inputs and outputs, a cortical neuron has a couple thousand inputs. You probably can't fit 1,000 parameters to 100,000 spikes. But you'd only have a few thousand stimuli if the neuron was linear. Now, there's a lot of evidence from synaptic physiology and dendritic physiology that neurons are not linear. There's NMDA spikes. There's a whole range of nonlinear effects in neurons. If they are nonlinear, then all of a sudden, instead of just only having a few thousand free parameters per neuron, you'd have maybe the square of that. And then it's pretty clear that your 100,000 spikes won't get you all that far. And on top of that, even if you have 100,000 spikes, a neuron isn't stationary. So in the early spikes will be different from those at the end. So you have extra parameters that you need to track the fact that they change over time. So that idea that we could reverse engineer every neuron and get at the full understanding might not work either. And of course, yeah, machine learning can somewhat help by allowing us to deal with many inputs and regularizing and allowing nonlinearities. But there's still that problem that it's unclear. If the neurons are not trivial, it's unclear if we can identify what the output is. Summary of my discussion of where we are experiment-wise is, I think we know relatively little about how the brain works. We know a lot of things about the brain, and we know a lot of super useful things about the brain, but I don't think we know much about how it actually computes. And I don't think data itself will solve that problem. If you could have the 100,000 spikes record all presynaptic inputs of all neurons in the brain, you probably still couldn't identify it. And so I think we need theories. And I just, so how am I doing on time? We're finishing in 5 minutes, something like that? Yeah. Okay, great. So I think we do need models. And I want to just briefly tell you about a class of models that I'm recently very excited about, which are models from deep learning. Now, what's special about models from deep learning? Well, deep learning is solving real problems. So the typical models that we have in neuroscience don't do anything useful. If you have a linear model of what a group of neurons are doing, we have figured out linear learning in machine learning a long time ago. So in that sense, we don't understand anymore about computation or machine learning by studying very simple functions. And deep learning is able to solve real problems. Linear learning isn't, and that's why I think this is a space where there's a lot of interesting questions. And the first thing I want to say is, one thing that makes neuroscience difficult is the heterogeneity. If you look at a group of neurons, every neuron looks rather considerably different from one another. And the early work in both the retina and V1 made it sound as if neurons were simple. No, it was like point detectors and it was edge detectors. Well, it turns out that if you carefully do retinal physiology, they care about movement and all kinds of other things. And Markus Meister is showing us how they're yet one level more complex every year relative to the previous one. And the same thing, huge heterogeneity of neural properties in already layer V1. The further up you go, the more complex it gets. So the first thing, so a big problem that we have in neuroscience is how to think about heterogeneity. And that is actually something where deep learning and in general cost function optimization is really good. If I give you a complex world, say the video from the camera that we mentioned earlier, and I give you a very simple learning rule, maybe extract the principal components of that, do Hebbian learning, do nonlinear Hebbian learning, something like that, you can produce a whole heterogeneous population of neurons based on that. And in that sense, it promises to solve one of the big problems that we have in describing neural populations. And let me highlight why that would be useful. So you could say those are just two duals. Now we have the representation, which is the tuning curve of every cell. And alternatively, we have a cost function and an optimizer of that cost function. Why would it be better to think about it in terms of a cost function and an optimizer? Well, the complexity of our tuning curves can contain any information we ever get from the world. The world hands us about 10 to the 7 bytes, plus minus a few orders of magnitude, depending on how you think about it. So by the time you do experiments on an animal or a human, that's an unbelievably big data set. And the representation in general is huge. If you take all tuning curves, which would be effectively all the synaptic weights in the entire brain, you'd need 10 to the 15 bytes, which is, and this information is huge relative to the size of the genome. Now, the genome upper bounds the complexity of the learning rules that you have in your neurons. So in that sense, and it probably massively upper bounds it because the vast majority of the genome doesn't deal with specific synaptic learning rules. So in that sense, we have good reasons to assume that the learning rule is way easier to understand than the representation. And I very briefly want to skip through that, the paper with Marblestone describes all that. I just want to add 3 things. I don't think it's as gradient descent learning is as unrealistic as people think. There's a lot of recent theoretical work that suggests that it's actually quite compatible with what we know about cells. We also speculate that the brain doesn't have just one cost function. It's not that you just maximize the number of successful guesses. You probably care about how your visual system can predict the auditory input and vice versa. So there's a lot of components that may change over time. And lastly, you have those specialized systems. And in fact, if you look at the deep learning literature at the moment, a lot of people start building specialized systems into their neural networks because it improves performance. Very briefly, what can we do? I think we can do generalization experiments. I think the reason why we failed so miserably at the microprocessors is in large parts because we use these complex behaviors. Now, the typical neuroscience experiment doesn't do a good job at getting at an atom of computations. Everything is involved. There's motivation and reward and sequences and boredom and all that. And in that sense, finding good behaviors that really separate one aspect of what the brain is key. Non-trivial data analysis. The brain is nonlinear and recurrent, and we use techniques that have been developed for linear systems, and we just use them because we can download the code and it's easy, but we need to do justice to the complexity of the brain there. And lastly, I think we need to build theories, non-trivial theories, and I think deep learning is a nice area to do that. Okay, let me skip through that. Thank the people who did it. Eric Jonas, who for some reason vanished here, did the whole microprocessor analysis, and then the rest was with Pavan, Ari, and Ugo, and those are our collaborators. And I want to add one last shameless promotion here. I recently wrote a paper called 10 Simple Rules for Structuring Papers. And if you're struggling with your early papers in your career, this paper basically breaks down all the components of how to write a paper. What does the first paragraph in the introduction do? What does the last one do? It's kind of that outline of how typical papers are. And certainly for students in my lab, I've been getting much better drafts since I make everyone read that. And with that, thank you. That's right. We can't go to the microprocessor guys and ask, give us your algorithm and then adapt them to brains. That's absolutely true. But at the same time, as long as we can't properly make progress at understanding, say, unknown microprocessors, it's very hard to argue that we can make progress at understanding brains. I mean, like, this is effectively a test case. So if in software development, no one would do proper software development without having test cases where you are, for these set of situations, this is what our algorithm needs to do, and you test if that works. And at some level in science, as science gets very complex, as in neuroscience, and arguably sociology, a lot of other disciplines, we need to start having test cases to know if our assumptions, if even under the assumptions that we make ourselves, we'd be able to understand the system. Yes, so gradient descent learning is super attractive because you can take any nonlinear function and in every step, and it allows you to step-by-step optimize that. And if you have a deep system, it also allows you to get close to a global minimum there. So in that sense, computation is very attractive. So starting with, or like Hinton has done a lot of work in that general space. The idea is, how could neurons do something like that? And there's a broad range of approaches people have proposed. During my PhD, I proposed that the apical dendrite of pyramidal neurons effectively computes the gradients. And there's simple, relatively benign physiological assumptions that would allow it to do so. There is, by O'Reilly and Hinton have worked a lot on these recirculations algorithms. So the idea is a little the following. You give an input to the network. It goes forward, percolates to that system all the way to the end. And at some level, each neuron stores what was going on there. And then what you do is you take the output and you clamp it to its true value. So you basically say, well, that was a lion that actually did bite me. And then, so that one, you force it to then stay on based on some feedback that you're giving. And then you allow the system to, and you have always, and somehow enforce symmetric weights, and there's a lot of local learning rules that would make the weights be symmetrical. And then you let it relax after forcing it to be the right value. And then if you look at that difference between the two of them, in such a system, that tells you the gradient. So in that sense, if you have a system like cortex, where you have one mode, maybe when the stimulus comes in, where it percolates forward, and the other one where you clamp it, then it would allow each synapse to calculate that derivative. That's another proposal. And then there's a set of proposals where you use neuromodulators to propagate information. Was that a successful thing that I just did or not? And in first order, you can approximate that. And there's a nice Tim Lillicrap, yes. Tim Lillicrap has, that was amazing that you could guess it based on that. Tim Lillicrap has a nice paper where it says, even if I choose the backwards going weights randomly, it can still get something that is rather close to gradient descent learning. Great point. Those are two completely different uses of deep learning. There is the machine learning for neural data analysis aspect, where I don't have a theory about how the brain works. All I do is I take data, say the inputs and the outputs of a neuron, and I want to somehow estimate that nonlinear function connecting the two of them. That is an engineering problem for which we can use deep learning. So in that sense, deep learning isn't a model of how the brain works. In that case, deep learning is a tool that we use to probe data. And then at that last point, now we take deep learning as a theory of that. So what we do is we train a neural network to maybe solve a vision task, maybe object recognition. And then what we can do is we can say, well, to which level is that neural network that we trained similar to the brain? And we may hear about that later. But that's really, that's the same thing is actually true for Bayesian statistics, which is an area where I was very active in the past. So in Bayesian statistics, you can have a model of the overall brain where I say to which level is what you as a human are doing, to which level is that optimal for solving a class of problems. And then there's the microscopic application where I say, well, I get 100,000 spikes. What's the best statistical estimate that I can have about the property of the neuron? And that's a Bayesian problem. And in one case, for one of them, it's a pure engineering problem, which is how can I best fit a nonlinear function to some data? Or what's the most probable fit that I can have here? And the other one, it is really a high-level predictive model. Yeah, and the right ways of doing that are different. So if what you care about is just that you can fix it, the right research may very well be if someone's depressed, let's just randomly cut into their brain if we and take good care of log that, or like reversibly cut into their brain, and then we may well discover. And a lot of, no, I mean, like, that sounds malicious the way I'm saying it, but arguably the vast majority of actual treatments in medicine are found like that. But I mean, brain stimulation works great for Parkinson's disease. We have no meaningful theories on how and why it should be working great. And just some mature neurosurgeon at some point of time tried it, and it worked, and that's great. I mean, like, not without some background, but suddenly without a full understanding of why it should be working. And so this research, if this is the hat that you're wearing, you really want to basically have hypotheses and about what's treatable and test that. If you want to simulate it, then you really probably want to solve the problem of how does a neuron relate to its inputs and have that for all of them. If what you want to do is analyze it at a computational level, which is usually done at a behavioral level, you don't even care about the brain. And arguably, a lot of people at that level say that neuroscience is a distraction. That if we want to be good at predicting human behavior, that the brain is entirely besides the point. That cognitive science actually has the correct language at describing macroscopic behavior. So each of these forces us into a different level. And but to wrap it back for the nature of models, if you want, at this level, you want models that are matched to the kind of interventions that you can have. At this level, you want models that are mechanistic. I think when I talk about deep learning as models, they kind of lie roughly here in the logic. But yeah, and let me explain to the students here why this disagreement here matters. So at some level, when it comes to the okay, but why this debate matters? At some level, the biggest questions that are behind our research we almost never ask. What is it? What does it mean for me to succeed as a scientist? What does it mean for me to understand? What is the level of description that I want to have? And personally, in different work of mine, I'm wearing every single one of these hats. And in that sense, and every one community kind of looks down on the other like 6 that we have on that slide in a really interesting way. Yeah, so I'm personally very I don't allow people in my lab to use the C word. It feels like a question that I can't properly write down. And personally, I have great trouble with theories of consciousness that are substrate dependent. It seems so. At some level, there's the information level theory, if you want, like the computational way of thinking about it, and then there's the mechanism way. And at some level, this is the level at which I personally ask those questions. But yeah, no, I don't think I can give a good answer to that.
Well, let's take a thanks again.